Hello there! In this lesson we're going to learn about the cell cycle. The cell cycle is a pattern of growth and division that all cells follow ever since the first cells billions of years ago. So we're going to focus on cells that have nuclei, which are eukaryotic cells, but there's a similar pattern that prokaryotes follow as well. So the purpose of the cell cycle is to enable for cell division, which produces new cells. And these new cells are going to be genetically identical to each other, which means that they have the same DNA. So if you're a single-celled organism, like a bacterium or any of those protists that live in the pond, you are going to be reproducing asexually in a similar process. You're going to need to copy the DNA that's in the original cell and then divide it up and end up with two new cells that they call daughter cells in the end. For sexual reproduction, you do end up with some mixing at the beginning in order to make that first parent cell, but then ever after that, it's the same pattern of replication and cell division with the exact same DNA again. So that's the process that we're going to be looking at more closely in this slideshow. All of our multicellular organisms follow this pattern, which includes, of course, humans and all the plants and animals that you run into on a daily basis. So you may be wondering why divide at all, or why divide at such a small size? Why not grow a little larger? All the cells that you've ever seen are all from under a microscope. They're too small to be seen with your eye. And so the answer to that is that it's a lot more efficient to have a small cell because of the way in which nutrients and waste move inside or in and out of the cell. Think of some oxygen that needs to get into the cell to be used. It's a lot farther to get inside to the center of this larger cell than it is in the smaller one, where it might be needed by, say, a mitochondria. Same with waste, a lot easier to get out of a small cell than it is to get out of a bigger one. The other thing has to do with the amount of surface area that there is. Now, of course, a smaller cell is smaller in all senses, but it actually has a lot more surface area compared to the amount of volume that's inside of it. So if you take a bunch of these little ones and put them inside the bigger one, you can kind of see this. The bigger cell can only diffuse in and out at the outer surface here, my terrible sketches, right? All around here. Whereas with the same volume or even less volume, you get a lot more surface area where waste and nutrients can be exchanged with the environment, right? So the smaller the cell, the more efficient it is due to the smaller distances it has to diffuse and the more surface area that it has for its size. Alrighty, so let's make a journey through the cell cycle. We'll start with our parent cell that's going to be copied and form some offspring cells. And you see there's two brightly colored segments that this parent cell is going to go through. The first phase is called interphase. And during interphase, the cell is going to grow, it's going to get bigger, and it's also going to copy its DNA. And the fancy word for copying is replication. So those two things are going to happen during the first segment called interphase. The second phase that we're going to go through is called mitosis. Mitosis is where we're going to divide up the DNA that was replicated into two groups or two identical packages that's going to make the new nuclei. And then the final bit is to split up the rest of the cell in a process that's called cytokinesis once all the DNA has been copied and divided. So the first step along this replication cycle is going to be interphase, where we're going to take our parent cell and it is going to go through some changes in order to get it ready for dividing. Now, some cells never leave interphase and never divide again. Things like nerve cells in our body, they develop to a certain point and then there are little chemical signals that cause it to stop. But if you're going to replicate, you're definitely going to need to make the cell bigger, because if you keep dividing your cell, it's just going to get smaller and smaller until there's nothing left. So you got to grow first, and then there's what they call an S phase, where the DNA is copied. You get a second copy of the DNA, and so you can see at the beginning of this S phase, there's only one copy of each chromosome, and at the end, we've got two copies of each chromosome. And then it's going to grow some more make it a little bit bigger so that it's ready to divide. Okay, so now that interphase is done, we have this nice big cell that's ready to go through the mitosis part of the cell cycle. And the first part of the cell cycle is called prophase, and some of the characteristics we're going to look at now. So the first off, you'll see the nuclear membrane starts to break down and disappear. So nuclear membrane, right? The second thing you'll see is that the DNA starts to condense into these chromosomes. And each of them is going to have 2 strands. So each strand's called a chromatid, and they're identical to each other. That's the part that was replicated during interphase. So you get 2 chromatids. The last thing you'll see sometimes, depending on the diagram, is these centrioles that are the organelle that's going to separate the chromatids from each other. And they'll be migrating to the poles, one to like the north and south pole here. And these little fibers here called spindle fibers will start to appear. And they're going to attach and separate the chromatids when the time is right. Alrighty, phase 2 of mitosis is called metaphase. And during this phase, you see that the centrioles have moved their way to the poles of the cell. You can kind of think of this like a globe on the Earth. You got north and south, and on either side you should have a centriole. And then those spindle fibers have now attached to each one of these chromosomes that are all lined up pretty much down the equator of the cell, or the middle. And so you can kind of remember that metaphase is when the chromosomes are in the middle. Both those M words go together, and it helps you to be able to identify them when you need to. So we're all set now to separate those identical pieces of DNA, the chromatids, from each other once you've entered metaphase. Step 3 in mitosis is called anaphase, and that is when those chromatids get separated from each other and are pulled towards the poles, right? So we got our north and south pole here. Now you got the chromatids that have been separated from each other. Now they're individual chromosomes, and they're all being pulled away from each other towards the centromeres on either side. And this should be an exact splitting of the DNA that was replicated in interphase. So now it's ready to make 2 new nuclei out of these identical sets of DNA. Finally, we've made it to telophase, which is the last of our phases of mitosis. And what happens there is sort of a reverse of prophase. You'll see that the DNA starts to unravel. You'll see that a nuclear membrane starts to form again. The centrioles and the spindle fibers, they let go and start to be taken apart themselves. And the cell's getting ready to split apart and become 2 new cells once this whole process is done. All right, so we finally made it to the end of the cell cycle. This last phase is called cytokinesis, and that's when the cytoplasm of that original parent cell is divided between the 2 new cells that are called daughter cells. And so the nuclei were divided equally through the process of mitosis, and now the last bit was just separating the 2 cells now into 2 identical daughter cells. And so each one of these will again start its way through its own cell cycle. It's going to be, of course, half the size of the original cell, and it's going to need to grow and replicate its DNA in order to divide again. So there you have it, the whole process, the cell cycle, interphase, mitosis, and cytokinesis, and you have 2 new identical cells. All right, hope that helped. I'll talk to you soon. Bye.